Hi everyone, my name is GARCíA, and I'm going to present my oral communication titled Bioinformatic Analysis Identifies Specific Expression of Non-Encoding RNA and mRNA in Pediatric B-cell acute lymphoblastic leukemia. And there is no conflict of interest in this talk. And the result of this research I have been published in these two articles. I will talk about the main results. And I will show the ideas that we conclude in these two publications. As an introduction, today acute lymphoblastic leukemia is the most common cancer in children. The pediatric B-cell all specifically constitute a heterogeneous and aggressive neoplasia in which new biomarker and therapies are required. Nowadays, we can use the next generation sequencing platforms and technologies to find biomarkers to improve the diagnosis or prognosis of the disease. Specifically, we can find changes in the gene expression or even in mutation in protein-coding genes or in non-coding RNA. We are interested in non-coding RNA, specifically in long non-coding RNAs that are transcribed longer than 200 base pairs that lack protein coding. It's known that this long non-coding RNA can regulate gene expression by different mechanisms. For example, by the regulation of the RNA splicing, as you can see here, or sequestrating the microRNA. The dysregulation of this non-encoding RNA has been described in cancer and can lead to oncogenic or tumor suppression as well. But what about the pediatric field? Today, there isn't a consensus of a clinical relevant non-encoding RNA signature for this disease in children. So the objectives of this work were perform a microarray analysis to study the non-encoding RNA and mRNA profiles in these patients, validate these results in external cohorts like the Cancer Genome Atlas or the Cancer Encyclopedia, analyze the survival of pediatric BL patients according to this expression. To accomplish this objective, we analyzed the gene expression of 42 pediatric BL samples and 4 healthy samples that were extracted from the bone marrow of the patients. We designed the microarray to analyze approximately 3,000 long-encoding RNAs and 2,000 protein-coding genes that are predicted to be regulated by these long-encoding RNAs. And we measured the gene expression and in order to identify long-encoding RNA that were differentially expressed. These results, on the other hand, we validated in different external cohorts we used data from the Cancer Genome Atlas that contains patient data and Cancer Cell Line Encyclopedia that contains cell line data. After the filtering and normalization steps, these were the numbers that we obtained for different samples, and we used it for the analysis of the gene expression and survival analysis even in different subtypes of the disease. All of these calculation and analysis were performed by our software in bioinformatic pipelines. So as a first result, we obtained a list of 170 differentially expressed non-encoding RNAs between BL patients in our microarray with the translocation 1221 and without the translocation 1221. The top differentially expressed non-encoding RNA mRNA pair was the TCL6, TCL1. And here, we obtain a high correlation in our microarray and other data sets for these two genes. So we felt that it could be very interesting for the clinical, and we published the article continue studying this pair. On the other side, in this next slide, you can see here a heat map the differentially expressed long-encoding RNA that we obtained comparing the BL samples and the healthy samples. We obtained a list of 20 long-encoding RNA that were overexpressed and 29 that were downexpressed. And here are representing this portal. Why did we decide to study this last pair that was published in the second article that I mentioned before in external data set? When we studied the gene expression of all long-encoding RNA and mRNA, we obtained the best correlation for this pair in our cohort and in different data sets. And also we saw that this pair was upregulated in other pediatric BL subtypes. So we decided to study and validate it in external databases. The first result that we obtained was for the cancer genome atlas. On the left, this box plot represents the gene expression of both genes, the AL1-333-46-1, that is the long-non-coding RNA, and the FIFN2, that is the protein coding gene. In blue for the PL samples, and in red for the TL samples. And here, these scatter plots represent the gene expression for both genes to visualize the distribution for every sample, that is every dot that you can see here. So as a result, the PL samples, as you can see here, show an increased expression for both genes, for the long coding RNA and from the protein coding gene in PL compared with TL. And there was only a significant correlation in this subtype in PL samples, not in PL samples. And this slide represents the same result, but for the Camphor line encyclopedia, for the Camphor line that we analyzed. And we obtained the same idea. The PL sample had a higher expression of both genes and a significant correlation. Next, to explain this correlation, we decided to study the genomic location of both genes, the long-non-coding RNA that is here and the protein coding gene that is here. And according to the gene enhancer database, these two genes share two enhancer or significant enhancer that could regulate their expression. So here we thought two possible regulations that can happen in this system. One of them is the long-non-coding RNA and the protein coding gene are specifically modulated in the all by the same regulatory element that are these enhancers you can see here, and the long-non-coding can regulate the FIFN2 expression. Lastly, regarding to the first long-non-coding RNA that we studied in the first publication, we performed a survival analysis with the survival data of patients that we download from or that we obtained for our cohort. And the patient groups were divided or split into two groups according to the expression of the gene, in high if above the median, or low if below the median of the expression. And on the group with a high expression of the TCL6, that is the long-non-coding RNA, show a better prognosis compared with the low group or the group with a low expression. That was for our first long-non-coding RNA. Regarding to our second pair that we obtained in our study, only in the case of FIFN2, we obtained a significant result. Those patients with high expression of FIFN2, the protein coding gene, associated with the long-non-coding AL1-233-46.1, show a better survival for those patients with a high expression. But we didn't obtain significant difference in survival for TL patients. So we thought that maybe this gene could be a specific biomarker for the PO subtype, for the B-cell subtype. We perform a Cox analysis, and we can demonstrate that the FIFN2 expression could be an independent prognostic factor associated, in this case, to longer overall survival. So as a conclusion, we identified at least 48 long-non-coding RNA that were differentially expressed between pediatric PO and healthy bone marrow samples, and at least comparing the translocation 1221, BL samples show a higher expression and correlation levels for this pair than TL samples in external datasets, specifically in the Cancer Genome Atlas. And the survival analysis demonstrates that patients with high expression levels of TCL6, our first long-non-coding RNA, and the FIFN2, our second protein coding gene, show a better prognosis than those with low levels. And lastly, this result can suggest that the long-non-coding could be a possible therapeutic target in the future, as FIFN2 could represent an independent prognostic factor in pediatric PO. I would like to thank the Gene Expression Regulation Cancer Group for their help, and all the funders that you can see here, and especially the Aula de Investigación sobre la leucemia infantil y leucemia for making this research possible. Thank you for your attention. If you have any questions, we will have to answer.